So this is what we've already been going through. Kind of a reminder. An allele is a version of a gene. Sometimes there's only one version of a gene, which means that it's gone to fixation, and any other version is has less fitness. So an allele is either from mom or dad. They sometimes have the same allele, oftentimes different. And of course, those alleles on those bands that we were looking at, those are alleles. So, which patterns can't belong to a child of this couple? So we've got dad here. Got dad here, there, and there. Got mom up here. And let's see here. Oh, I see. So which one is it? C. Okay. Okay. Okay, has to be D. So this allele right here we have what's called a double band. That means we have the same allele. Same thing up here, we've got a double band. So mom and dad share this same allele. Here, mom doesn't have this, but dad does, but it's a double band. So if you had a double band there, it means the child got both alleles from the father. You can't have that. So it's D. So what kind of cell is this? Eukaryotic. Okay, good. Now remember, all eukaryotes have a nucleus, and they often all have mitochondria and all this other stuff. Now remember we talked about a prokaryote, they don't really exist. They're either bacteria or archaea, neither of which have a nucleus. So if we were talking about a bacteria, it would be generally a lot smaller. Okay? If it was a virus, it'd be smaller still. Except for some new viruses that have been found that are gigantic. Who are these guys? Okay, good. 1953. So complementary base pairs. Okay, and you guys got it. Good. So, which enzyme is used for this? DNA polymerase. Okay. So it adds the complementary nucleotides to a single strand here. So that's how you can actually copy a cell when it needs to be copied. So what procedure will they use to amplify a small amount of DNA to a large? Okay, PCR. So, why are STRs so useful? Well, here's the repeats. This person has 3, 4, 7 repeats. Can we differentiate this person from someone else? Someone else might have 8, or 9, or 3 repeats. And so we can use these STRs to figure out if someone is actually a possible suspect or not. Here's gel electrophoresis again. So we take these different fragments or samples from different people. So one sample per well, put them in, charge it. The shorter fragments go faster, further down. The longer move more slowly. So a chromosome is, of course, one long DNA molecule. Each chromosome, one long DNA molecule with millions of bases or nucleotides. Now, this is a homologous chromosome, of course. So, one from mama, one from papa. Here's the gene. You want to duplicate. And then genes make proteins. That is, a gene is a stretch of DNA that is translated into a protein. Now, a gene really doesn't do anything per se. All it does is it is used as a template for making the protein that you want. Okay, so we've done a little bit of this, but let's take a look. Each of these is an amino acid. A protein is essentially a string of amino acids. Depending on which amino acids are in which place, often determines how the protein is folded and exactly what are the active sites. So what happens is the gene on the chromosome is translated into these amino acids. Now, okay, so here we go. This is the nucleus. Okay, all inside. The DNA is inside the nucleus for a eukaryotic cell. Bacteria, it's not. There is no nucleus. So transcription and translation happen with the bacteria together. Here they're separated. So what I mean by transcription is that this is a cytoplasm. That's the outside of this. That's not the outside of the cell, but it's the outside of the nucleus, where all the mitochondria are. And inside is the DNA. So what happens is the DNA has to be transcribed into a single strand of RNA. So it goes you go from DNA to right there, RNA, and this is mRNA, which means messenger RNA. The messenger RNA is an exact copy of the DNA, but it's a complement of it. Okay? So, at some place you have, let's say, a T, it's going to be, for RNA, it's going to be, we'd say it's normally going to be an A, but actually it's called uracil. It changes a little bit in RNA. But the main thing is you go from DNA to RNA, and this is called okay. Well, let's get this over here. Okay. Now, this messenger RNA, which is a complement to one of the DNA strands, it moves through this pore. So there's ways to get things in and out of a nucleus, and it's a protein pore. And here's the mRNA. And it's now in the cytoplasm. Now, it turns out here are these amino acids, free amino acids, and we have this ribosome. Now, the ribosome is basically an RNA machine, and it's made of RNA. Therefore, what it does is it takes the amino acids using the mRNA as a template and synthesizes the protein. And that's called translation. Okay, and then we go in the ribosome. This is tRNA, and then this goes to protein. And we'll say right here. So we go transcription to translation. Transcription happens inside the nucleus. Translation happens in the cytoplasm. Amino acids are essentially peptides, and they're strung together in a long polypeptide chain. Like this. And this is what the protein actually is. Okay. So this is the basic process by which DNA is turned into a protein. And proteins, remember, do all the work. There are structural proteins. There are proteins like enzymes that handle chemical reactions. There are signaling proteins. Everything that is in our body is all proteins. And those proteins, sometimes they can be long chains, sometimes they can be short, sometimes they can be isomers or of each other, slightly different. And of course, this is where variation comes in, ultimate variation in terms of mutations. Okay. And actually, this should be a RNA in here. And that's transfer RNA, and we'll get to that now. Okay. Now, transcribe. Kind of like a court reporter. So we're taking some code that has information in it and turning it into a way that it can then be used. Translation, and in this case, translation is making amino acids that then become proteins. Let's say from one language to another. I want to do one thing. Okay. Okay. These are nucleotides. It won't work. Okay. We'll say that's an A. Say this is a G. This is a C. T, T, A, G, G, A. Okay. Now, you need 3 nucleotides to make an amino acid. One won't do it. So, here we've got 3 nucleotides. It's called a codon. And this amino acid AGG, or this code, will make an amino acid, whatever it is. Okay? And Remember, these are peptides that then are strung together in proteins. So we've got some amino acid here. Here we have another amino acid. And here we have another amino acid. So we've got 3 amino acids stuck together. Each 3 is a codon. You need 3 to make an amino acid. Now, occasionally, I showed this earlier, where we looked at reading frames. So a reading frame is always going to be a 3 DNA letters long. If it stays a reading frame, that's good. If we were to actually start instead right here, let's say we had a mutation, then we'd end up with a different amino acid or maybe a stop codon. And probably everything downstream of this would all be gobbledygook. So now let's say we do manage to stay in the reading frame, like right here. But let's say we've deleted 3 amino acids. And now we go from here to here. It means that we've still got amino acids that make sense. They're not nonsense proteins, but they're different. And so it's a mutation that might change function, it might not. Depends. Okay. So when Watson and Crick discovered the structure of DNA, they didn't know how to read it. They knew they had the structure of it, but they didn't actually understand about codons, like triplet base pairs, how that translated into RNAs and then into proteins. And but then they found out, in fact, it was Crick who actually found out all those different things. And people thought, well, that's pretty much how things happen. You have different DNA, you get different amino acids, different proteins, different functions. Some proteins work differently than others and might be favored in one environment versus another. But they didn't know anything about what's called gene expression or regulation. That means you're regulating how much DNA is turned into proteins or how much protein you're making. Sometimes you want a little bit of protein, sometimes you want a lot of protein. Let's say an enzyme like lactase, which breaks down milk from a polysaccharide into a monosaccharide, right? Which is a single sugar, glucose. Now, until, I don't know, maybe 10 years ago, we knew virtually nothing about gene expression and regulation. Theoretically, we thought it might be important, but we didn't have the tools to actually look at it. And then slowly we developed. And so we're kind of in a renaissance now, where we're finding out things that we never knew before. Now, one of the things that gene expression does is it uses genes to differentiate cells. Now remember, each cell has a complete complement of the whole genome. All 23 homologous chromosomes. So it's not like one cell only has a few for it and they all have the same thing. And part of gene expression is how you get cells to differentiate. So in a muscle cell, right here, certain genes will turn on and off. And you'll end up with a muscle cell. These are all little, in each cell there's a nucleus. Versus a neuron, let's say, where they look different, they function differently, and each one, of course, is made from different proteins. So one of the things that gene expression does is it tells which kind of cell is being made, and especially during development, you want neurons in certain places and muscle cells in others. And that's kind of what one of the things it does. So gene expression is actually where everything is actually happening now. I guess it's late. Why don't we take a little break, and then we'll get back to this. Oh, I'm going to put up another top hat, too. Okay, so this is where we kind of left off. So we've got our genome, and with all the base pairs, and those code for proteins. And then it turns out that that's only a small part of the story. The big part of the story is gene expression. The first thing that genes do is they help cells differentiate, let's say during development. So if you look up here, it's not really an instruction manual per se. It's always going to be a gene by environment interaction. The genes interact with the environment, and that affects how genes are actually expressed. But let's say you have muscle, then you've got genes that are going to affect the muscle, the heart, which is a muscle also. Let's say building muscle. So you start working out, you're going to have gene expression there. The same thing goes with, let's say, eyes, that kind of stuff. Now, to get a point across that it's always a gene-environment interaction, there's a small fish called the zebrafish that is kind of a model organism that people study. And they know the genome, they know a lot of things about it, and they look at development in it, a bunch of different things. It turns out that the heart for a zebrafish is developed much like a heart for us, for both vertebrates. In the beginning, what happens is it starts out, a circulatory kind of system starts out in a very rudimentary form. And the vessels form, and then the blood plasma is circulating, and it causes shear forces, like it bumps up against the walls of vessels, and that's actually what causes the heart at a certain place to form. without that circulatory system, a heart wouldn't form naturally at all. So in this case, it's a cellular environment and a genetic environment. It's not the outside environment. So environments are at all sorts of different scales. Similarly, we talked about wolves before, and if it got warmer, colder, whatever. We've obviously got genes that code for, let's say, perhaps heavier fur if it's getting colder. But remember, there's always a summertime and a winter time, and during that time, like during the winter, the genes are going to be expressed so that they have denser fur. And then when it starts having longer days and warmer, you're going to end up with a summer coat that's much lighter. And that's all because of gene expression. Okay. Now, we're diploid organisms, you and I. That means, well, what does that mean? Anybody in here know what a diploid organism is compared to a haploid? Do you think sperm and eggs are haploid or diploid? I'm just wanting you. What do you think? Haploid or diploid? Okay. Dip, what does that sound like? D, 2, diploid, that's 2. Sperm and eggs are haploid. Okay? So, our gametes are haploid. They each have 23 chromosomes. And then when they fertilize, the egg is fertilized by the sperm. You end up with a diploid zygote that starts to undergo developmental dividing. So if we have 4 different proteins, and we have, let's say, protein A. This is from muscle cell. What's the difference between transcription and translation? Anybody? What do you think? What's transcription? Does it happen inside the nucleus or outside the nucleus? Inside? Okay.
Translation.
Cytoplasm. Good. Okay. So, transcription, you're going from the DNA and you're transcribing it to messenger RNA. And then the messenger RNA is filtered out or ferried out of the nucleus into the cytoplasm, where it's then translated into amino acids and proteins. And proteins are made of amino acids that are strung together in a certain order. Okay, so here we have transcription. You go to RNA, then to DNA, then to a protein. We also have protein B, less of it. And then C. So we have more expression of the protein for protein A, less for B, and about in the between for C. D in this case is off. In other words, that's not being transcribed or translated. Now, let's say we have a neuron. Neurons are, well, we have a lot of them in our brain. We also have neurons that branch out from the spinal column to all our different muscles and various things that control our body. Again, we have A is being made, B is off, C is on, and D is on. Now, if you look at where did I put that? If you look at C, in terms of muscle cell versus neuron, less C protein is actually being made. So they've got they've got genes that code for this protein, C, but the amount of protein all depends on how it's actually expressed or regulated. So we've got this promoter region, and then instead of an on-off switch, which would just give you the same amount of protein, let's say C, it's more like a rheostat. And a rheostat is like a dimmer, right? Just like this. It's a matter of dimming or turning up. The more you turn up, the more protein is made of that specific kind of protein. Now, the gene actually codes, of course, for amino acids, which then make up a protein. What is this? Okay, so there's anybody in here who's lactose intolerant? Okay. Now, for mammals, when do they drink milk? What age? Young, right? Babies. Okay. And then, for most mammals, of course, after they get weaned, they're not drinking milk anymore. And in most cases, this lactase, which is an enzyme that breaks down lactose into sugars, it gets turned off. So, because where is an adult mammal going to get milk after that? It's not. Now, the thing is, with humans, in cultures that were pastoral, where they had sheep and goats and cattle, things like that, they had milk. Most people in the beginning, of course, couldn't really digest milk very well. But there was a mutation, not in the lactose gene, or the lactase gene, but in the promoter region, and which actually kept the lactase gene on. In other words, it was producing lactase, and that individual could actually drink milk. Now, making a protein, in this case an enzyme, is not being used as a waste of time. So normally that individual would have lower fitness. But in this case, if there's milk around, no one else is drinking it, that person has an advantage. Milk's a high energy food source. And so, if you remember evolution, in this case, having that mutation where you've got cattle, and let's say it gets, there's not very much food left, and you start drinking milk, everybody else is getting sick from drinking it, and you are the one that survived. You survive, the other ones don't, you leave more offspring, and then over time, this mutation spreads in the population. So wherever we've actually had, let's say, a dairying culture or a pastoral culture, this kind of mutation has shown up. But it's shown up differently. It's always in the promoter region, but for example, the people in the Middle East and Northern Europe have a mutation in the promoter region that keeps the lactase gene on. But in Africa, there's two other populations that had different mutations in the promoter region that kept it on. So we know at least 3 times, in this case, all with kind of pastoral cultures, that the lactase gene is on because those people have access to milk and they can now digest it. So this is how it actually works. We've got a homologous chromosome up there. We've got this coding region. That's the DNA. It's then transcribed into RNA and then into a protein. And that protein is lactase. It's an enzyme. If you take the lactose molecule, it fits into this enzyme. The enzyme has active sites. And once it fits in, it actually breaks apart a glucose and a galactose. Both are different sugars. The glucose is what we need for glucose metabolism, which is what our body mainly uses. Now, if in fact the lactase gene, if sucrose comes in, it just splits apart sucrose, which is a disaccharide, into 2 glucose molecules. So this is actually how this gene is regulated. Now, also, it's not just regulated amount in terms of being on or off. It's also in the amount of galactase that's actually being produced. So here's our transcription. We've got, here we go. Okay, so here's our mRNA. That's the messenger RNA that is now in the cytoplasm. It was made in the nucleus. And if you remember, it's a single strand, but you can make complementary base pairs out of it. So with an RNA, you have a C, a G, a T, and then instead of an A, you actually have a U. And you can make amino acids from that. It tells you. So if you have 3 bases in a row, you get a certain amino acid. If you have different bases, you get a different amino acid. And then those amino acids, of course, are strung together. So here we have this promoter region up here. And now what we're doing is we're, let's see. Oh yeah. Here's these RNA nucleotides that are being strung together. Oh, this is, okay, my mistake. This is inside the nucleus. Looks more like a ribosome. Okay, so we're making mRNA that's going to be then ferried out into the cytoplasm and make the protein. We use another enzyme here called RNA polymerase. Anything with ASC is an enzyme. It's a protein that basically helps carry out chemical reactions. Okay. So the central dogma, it always goes this way, just so you know. It goes from DNA to a transcript, then translated into a polymer of amino acids, which makes a protein. You can't go from a protein. Now, proteins do the work, like an enzyme. An enzyme helps carry out a chemical reaction, usually at a lower effective energy than you would need otherwise. And so all the proteins that do these things, they're all proteins that do the work. DNA by itself doesn't do much of anything. It's just a template. The RNA, of course, is just strictly a template 2 that's made from the DNA. So you can never go from, let's say, a protein and then re-engineer an mRNA or a DNA from that. It only goes in one direction. Now, with RNA-based organisms, like some viruses, some RNAs actually are able to do chemical reactions on their own. So instead of going and having to make a protein, a lot of them catalyze reactions as well. But for the most part, it's always essential. Okay, and then there's translation. Now, the translation is happening in the ribosome. Let's see here. 1 second. Okay, and here's our mRNA that's being read. And remember, a codon is 3 base pairs long. So here we have AAA, that makes a certain kind of amino acid. UAU, that makes another kind. Remember, U actually replaces T with an RNA, but it's the same thing. And so now we're making this long chain of amino acids, which is the polypeptide chain, which is actually a protein. Eventually, it'll come to a point where it hits this codon. It's a stop codon. It says, oh, stop transcribing or translating. Now, there's another RNA. It's called the tRNA, transfer RNA. And what's happening now is you're actually transferring the RNA. If we look at it correctly, here's our codon, UUU. Its complement is AAA, which is the anticodon. This is the mRNA. Here's the tRNA, and this is where it actually, you put together the amino acids. So the tRNA then translates into a specific amino acid. There's a binding site, and there's this region of complementarity. And in this case, it makes an amino acid called phenylalanine. There are 20 amino acids total that humans use. There's actually more than that, but we don't use those. So tRNAs are actually the interpreter of the genetic code. Now, the genetic code, so these are the various different amino acids. There's 20 of them here. This is basically the universal genetic code. All organisms on this planet have the same genetic code. What does that imply in terms of life on this planet? Would you say life on this planet is all related to each other or not? From that evidence, right? Yes. So now, if we had some other genetic code, then it would indicate perhaps that there was another life form, that life arose more than once. Now, it could have been that, of course, life did arise more than once, but the only thing left over are our life form. Or it could also be, of course, that there's another life form on this planet that we just don't recognize as life. So this genetic code that we're looking at here, remember, it's always a matter of 3 bases long. So this is the first base. If we look at this, Actually, okay. So the first base is this. That's the U. In this case, this is the C, and then A. Okay? Now, the second base on this chart is the middle base. And here we have the C. Here we also have a C. They're all the same. Oops. Okay. And then finally, there's a third base, which is the last one. Now, any changes in the first two bases, points on the base, in other words, base 1, base 2, you're usually going to have a change in the protein. But the third base is redundant. You can actually often have, doesn't matter what the letter is, but you'll end up with the same amino acid. So if we look at this, we've got this third base. Even though it's UCAG, all the different RNA peptides, you still get arginine, which means that it's 4 degrees degenerate. No matter what's going to happen, you're going to end up with the same amino acid. Okay. So how does DNA actually affect the traits? Well, in terms of taste, we've got these taste receptors. There's one missing here. First of all, there's bitter, there's sour, there's salty and sweet. In the middle, there's not many taste buds. And there's also another one called savory, which is kind of a meaty flavor, as it turns out. So all humans have these, but taste receptor peptides vary with populations and with individual people. And I'll show you what I mean. Does anybody in here not like, what is it? What? Broccoli? No. It's, you eat it with Spanish food a lot, or Mexican food.
Spicy?
What?
Spicy?
Cilantro, yeah, exactly. Okay. Anybody in here not like cilantro? Okay. Now, a lot of people, they're fine with cilantro, but some, it tastes kind of metallic. They don't like it. It's not because they're being picky. It means that their taste receptors are a little different than other people. There are similar kinds of things where certain populations can taste things, certain things better than others. Now, this is all because of the way the DNA code is and how it codes for certain amino acids, and how you can get a difference in taste bud receptors and how many you have in certain places. So here we have what's called the hemoglobin gene. Hemoglobin is what carries our oxygen in our red blood cells. The hemoglobin gene, these are all the different AT, CGs, all that kind of stuff. Sequence, you have a nucleotide. You start out with, let's say, TCT, that's serine, AAG is lysine, GAG is glutamine, GAG is also glutamine, and so on. Well, that's the normal sequence. At the center of the hemoglobin gene is an iron molecule that actually carries, it has a high affinity for oxygen. Okay, now, if you notice here, let's see, we've got this guy here. Now, this is not a normal red blood cell, and it doesn't have a normal hemoglobin molecule. It's got a mutant. So in this case, though we went originally, we have this GAG, go to glutamine. In this case, it's been changed through a mutation, and the A has been switched to a T, and now it's coding for valine, or valine. Now, it turns out that having only one valine, so we have a homologous chromosome, one from mom, one from pop. If, let's say, only mom gives us a valine, this mutant, and dad doesn't, it means individuals who have this configuration are resistant to malaria. If they don't have it, they just have wild type up at the top, that means they're susceptible to malaria. Unfortunately, if they have the two mutants together, one from mom, one from pop, we end up with someone with sickle cell anemia, and who really has, usually they die younger, they don't get a chance to breathe. It's a heavy price to pay. It only really pays in an area where malaria is endemic, and those who have only a single copy outlive all the others. So this is, and that's exactly what happens with this individual right here. They, this valine here, they both get one from mom and one from pop. Now, if you get only one from mom and then a wild type from pop, you get pretty much a normal red blood cell with a normal hemoglobin in it. Slightly different, which makes it less resistant to a malaria parasite. Okay, GMOs. Anybody in here afraid of GMOs? You know what they are?
Genetically altered modified food.
Mm-hmm. They're called genetically modified organisms. Now remember, humans have been breeding organisms for years and years. That's how we end up with that corn over there. Farmers pick corn. There's always variation. They always planted corn with bigger kernels because it was more food. And that's called selective breeding and artificial selection. Well, there's also, nowadays you can actually put genes into an organism that weren't necessarily there. So you have a modified goat, you have a genetically modified corn, and it doesn't take as long as selective breeding, but you get up basically the same result. I have to change these. Okay. So, which is the right answer here? You know what a photoreceptor is? What does that sound like? Photo? Which answer is correct? A. Okay. Now remember, what's the name for the dimmer switch upstream that actually regulates gene expression? Starts with a P. Promoter. Okay. What's the two main steps that lead from a gene to a protein? The translation. Good. Okay. If we insert, true or false? If we insert a human gene, comes from humans, into a bacterial genome, the correct protein won't be made because the genetic code is different between bacteria and humans. Is that true or false? Do we share the same genetic code as bacteria?
Yes.
Okay, so this is a funny way to actually write this, but anyway, this is false. You can take a human gene, put it into a bacteria with a promoter, and get that bacteria to produce whatever that gene produces in terms of protein. Now that's what a genetically modified organism is. So, and we actually do this. Okay, so it's called recombinant DNA technology. For example, we have insulin. Now, previously, before this technology, we got insulin from pigs, and I think horses too. It's the same insulin, but there are some slight variations, and some people ended up having an allergic reaction to, let's say, pig insulin. So instead now, it says horse blood, but it was mainly pigs. And insulin, of course, is a protein that helps glucose, facilitates glucose metabolism. And now we've actually put the human form of insulin gene into a bacteria. Okay. What's happening now is, of course, bacteria is small, but if you have a huge vat of bacteria with this gene in it, you can make tremendous amounts of insulin. Human insulin, not horse insulin or pig insulin or anything like that. It's cheaper to make, get more of it, and it's the right kind of insulin. Now, when you take a gene from one organism and put it into another, it's called a transgenic organism. So the insulin-producing bacteria have a human gene in it. They're now transgenic. And that's what transgenic is. They have a gene from another organism. Of course, it doesn't seem right that we're doing this, but it's fine. And the insulin that, in this case, we're producing lots of it, is human insulin. Okay. So we can do this a lot. We can have other organisms make things that we want, not just bacteria. So, for example, antithrombin is a protein that helps prevent blood clots. People who have a mutation then have a problem in terms of clotting. Okay? So, this protein is made in the liver, mainly expressed in the liver cells. It releases antithrombin into the bloodstream, and it allows, it prevents the blood from actually clotting when it shouldn't. So there's some people who got a mutation. You can see how this would be a problem. They might make clots that would travel in the lungs into the brain and kill them. And in the past, the only antithrombin we got were from blood donations. But it turns out goats produces a massive amount of the protein if you put that gene in a goat, in the transgenic goat. Where do you think this antithrombin is found in terms of the goats?
Milk.
Milk? Yep, exactly. So, now to make this happen, remember, if you just put the gene in, without the promoter, nothing is going to get made. So you actually have to create a hybrid gene. You take a goat promoter and you attach it with the antithrombin coding sequence. You actually inject, let's see here. Okay, right there. You can see it's a very, very, very thin pipette, like a needle, and they're actually taking out the nucleus and replacing it with another one with the correct gene in it. And then you get the antithrombin. It's a human antithrombin. You get it from the milk, and it's easy to separate it out. We've been making all sorts of transgenic organisms. It turns out this jellyfish on the left is fluorescent. It lives in the deep ocean. There isn't much light. It's probably mainly used, I think, for sexual selection for reproduction. But someone realized they could actually attach a fluorescent protein into other organisms, into certain different types of cells, and make them fluoresce as well. So here we have, let's see. This is a neuron. I'm not sure what this is. And we can actually look at pathways. We can not only tag cells, but we can actually tag different proteins in cells and see how different pathways, different genes are expressed. and how different cells actually operate by using this technology. Now, transgenic organisms are used for all sorts of stuff, medicine, research, agriculture. You can engineer bacteria to produce like a growth hormone for cows that make them produce more milk. It's called the BGH protein. And what's happening here is you actually take the BGH gene from the cow and put it into a bacteria. And then the bacteria can serve as factories for, oh, come on. Okay, now the thing is, you're making BGH for the cow, because that way you can take it, inject it into the cow, they'll make more milk. You're not actually changing the cow, in this case. The transgenic organism is actually the bacteria that now has a cow gene in it. You think anybody in here, you think it's maybe bad to drink GBH in your milk? Anybody not want to? Okay. So far, it doesn't look like it's a problem. It could be down the line. People who drink lots of milk, let's say in the future, might, let's say, grow a leg out of their neck or something like that, right? So you'll be safe, everybody else will look funny. There's been a lot of questions about genetically modified organisms. I mean, it seems like the first thing that people not normally do is they're afraid, oh, it's genetically modified. Now remember, we've been modifying organisms for a very, very long time. We've just been doing it with a different method, which is selective breeding. In many cases, you get the same sort of stuff by genetically modifying things in a shorter period of time. The genes they put in was they inserted a gene that makes a natural, that makes a plant resistant to an herbicide, like Roundup. So if you spray it, it's not going to die. And so they put this kind of anti-herbicide in it, and they then plant it, they spray the field, get rid of all the weeds, and it grows, and everything is this genetically modified form. There's a question, of course, well, is this good for you? It doesn't seem to affect anything. We don't know. I mean, we know for the most part. There doesn't seem to be any bad effect. The one thing that's a little problematic, I think, is plants hybridize with other species of plants all the time. And you could possibly see how a gene that you put in to a plant would then spread to other plants, because when they actually pollinate each other, most of the time it doesn't work, but sometimes it does. And so you can see how a certain protein is being made by a bunch of plants that are now resistant to herbicide. That probably wouldn't be such a good idea. Actually buy in a store. When they genetically modified the corn, they put different genes in. Here we have rice, and it's been genetically modified. It's called golden rice. It produces, there's been a gene that's put in there that produces beta-carotene, which helps us make vitamin A. there are a lot of populations in the world that have vitamin A deficiency, and so we're now producing golden rice, and it's actually helping those populations, reversing the DNA, the vitamin A deficiency. Most people, some people might say, oh, maybe it's dangerous. It's been tested with all sorts of different organisms, in terms of, mice eating it, goats eating it, humans eating it. We don't see any evidence whatsoever that it's not healthy for a person to eat. And it actually increases vitamin A uptake and creation of vitamin A. Now, there's been some easier ways to insert a gene into a chromosome now. With a lot of plants, you can use this gun that shoots the particles with the right gene in it into the plant. So it's cartridge-containing DNA-coded microcarriers, and it pretty much works like any sort of a gun. Same thing with human, a gene gun. So there has been some work on, at one time it was thought that we could use gene therapy. Someone has, let's say, the antithrombin mutation. They're not making antithrombin. Maybe we could put in the correct gene into the human. The thing is, actually getting it into the right area in a chromosome might be difficult. You might, let's say, you might end up in some places right in between the middle of a gene and cause problems. So there has been some recent success after many, many, many years of trying for gene therapy. But in the beginning, it oftentimes didn't work very well. It's starting to work better now for certain diseases, certain genetic diseases. Whether we'll actually in the future be able to, let's say, modify a human being by shooting a gene into them in large quantities. Maybe they'll shoot a gene into me that makes growth faster, and all of a sudden they grow, right? That might be good, it might be fun, I don't know. I hate going to concerts where everybody's taller than me and I can't see over them. And it gets old having to kick them in the back of the leg, get out of the way. So anyway, it might be useful. Now, the trouble also is that, so you've got this corn. All the corn plants with this new gene in it are all grown from identical seeds and a whole field of identical corn. There is no variation there whatsoever. None. You go from one corn plant to the next, there might be a few small mutations, they won't have much effect, they're basically the same. Now, in terms of diversity, what do you think's happened with the diversity of corn? It's gone down, right? It's got one kind of corn. Right now, anybody in here like bananas? Okay, some people do. Anyway, the standard banana that we eat, there's a whole bunch of different kinds of bananas. The ones that we get here They don't bruise, they don't bruise easily, they travel well. They taste okay. There's others, kinds of bananas that actually taste better, but they don't travel well. Well, there's a new disease that's wiping out the standard banana. Everybody's, of course, planted only the standard banana because that's what the rest of the world wants. You can actually move it from place to place. And they haven't found any way to develop the standard banana with resistance to this kind of pathogen. So, yeah.
Has the standard banana already been genetically modified?
No. Well, it might have been. But it's pretty much like selective breeding. They've been basically, they've taken the standard banana and made sure that it's almost identical to each other, whether it's selective breeding or genetically modified. So there's a question now whether the standard banana is actually going to be wiped out. They've been trying all sorts of different things. Nothing has worked. Certain areas have lost their whole banana crops, and they never come back. I mean, they're dead with this pathogen. So one of the problems with an organism with no diversity, it may be really good growing it, let's say this banana, in the right conditions. It has better yields and all that. But if something comes along like a pathogen or let's say change in the climate or anything like that, there's so little genetic variation that they're usually susceptible to change in a way that's not good for the plant, let's say, or the animal. So that's been one of the problems, whether it's selective breeding or genetically modified organisms. For a long time, in terms of wood production, they've been planting kind of, a clonal crop of different kinds of pines, let's say Jeffrey pine. They grow fast. They can be harvested in 40 years or 50 years, something like that. And they're great, but they're also susceptible to, let's say, pathogens as well. So here's let's say, a corn, rows of corn, healthy on the left. On the right, maybe it was a drought, maybe it was a pathogen, whatever it is. Since all these corn are almost identical to each other, anything that comes in, and if they don't have natural resistance, they're going to have a problem. Okay, good. That's that. I'm going to open up Top Hat right now. For this section, and we'll get a little bit of a break. Okay.